Hi, my name is Celine, and today I'm going to present my report proposal of the metagenomics studies of honey. The contents here include the introduction of the research background, the problem statement, significance, and objective of this honey study, the literature review of metagenomics, where it will provide a more comprehensive review of it, the research methodology, where it includes the experimental design, expected results, and the Gantt chart. And last but not least, the reference that is being used to complete for this report proposal. For the introduction part, the green honey has recently been gained attention among the public and researchers due to its special green color when compared to the additional market standard yellowish brown honey. And green honey has been found in several areas, including in Philippines, Greek, and even at Kudat, Sarawak, Malaysia. However, the product's quality and authenticity of green honey has been doubted, so it is important to carry out the study of green honey to ensure that the consumer's health and also collect or consolidate information of the green honey metagenome. The microbiome of green honey can also be identified using metagenomic shotgun sequencing that has been carried out by the researcher of OWO. The customers will be able to get a better understanding of the nutritional value of the honey when they purchase, if they find that this information of green honey is interesting and important for them. The problem statement here is the lack of metagenome and genetic information of green honey has brought some challenges for the future research. This is due to the properties, health benefits, and sources of green honey that has not yet been identified by researchers. So the metagenomic approach is ideal for this study, but it's also raised several issues such as the lack of the universality in the primer and the low resolution of genetic markers. It is significant to carry out this study because the green honey study is still remain not much, so it is a future potential to be studied by the researchers, and it is important to provide a very clear methodology of metagenomic analysis on the green honey, and it can help us to understand the green honey composition, whether it is nutritious to human health or it will bring negative effect. The objective of this report proposal for the metagenomic studies of honey is to compare the microbiome activity of green honey with conventional honey such as tualang honey, gelam honey, acacia honey, and manuka honey. Secondly, the samples of green honey are collected to identify the source of the origin of the honeybees related community in Malaysia. Last, to identify the gene that codes for the green pigments in the green honey, whether it is obtained from the chlorophyll, algae, or other potential substances. In the literature review, the metagenomic research, it attempts to determine the whole collection of genes within the environmental sample, and it can analyze the biochemical activities and complex interaction. Besides the large scale of the shotgun metagenomics, there are also some small scale approaches such as the 16S RNA-based surveys. And the metagenomic study has been carried out, and application applies on a geographic tracing, the quality assessment of the honey, the utilization of the pollen DNA as a sampling means to detect the floral composition. Next, the honey production activities, the honeybees' complex relationship formed with the species across all domains such as plant, viruses, bacteria has been carried out, and it has also been used for making metagenomic tool for assessing the bee ecological niche that has been done by this researcher. Secondly, to trace the geographic origin of the honey, so we found that there is a mislabeling of the geographic origin of the food in the worldwide, including in the honey market. So this not only compromises for the confidence of the customers concerning the authenticity of the green honey, it is also raised several health concerns as the unknown origin of the honey, where it contains any antibiotic, toxin, any irradiated pollen from the flower, or even alkaloids with the potential to cause organ damage to the human. So the metagenomic data from the honey sample is important to derive those sequences and apply a backpropagation algorithm to determine the geographical provenances for the honey sample that has been done by the researchers from Shaolin Liu. There are two types of common analysis of honey metagenomics that's been carried out, which are the metagenomic shotgun sequencing and high throughput sequencing. In the metagenomic shotgun sequencing, the application that's been done including honeybee-derived environmental signatures and the honeybee hologenome complexity that's been carried out by the researcher named Bowo, and a multi-kingdom honeybee-derived environmental DNA signature from Bowo as well. And Galanis, this researcher has also studied the bee foraging experiences, microbiota, and pathogens using metagenomic shotgun sequencing. In HTS, which is the high throughput sequencing, it has been applied to pollen or honey identification to determine the floral composition and identify the geographical origins of different kinds of honey. However, there's also some limitations in the metagenomics of honey from the articles that have been identified. Firstly, the taxonomy identification process is highly reliant on the records or information available from the formally described species. Second, the particular taxonomy classification tools for the identification of honey-related species is still lacking, and the metagenomic classification tools are rarely optimized for particular application. For example, it may cause the present genomic library to have some construct bias happen. The sequencing of the microbiome is also more labor-intensive as it requires the culture of the individual species and the extraction and the sequencing of each culture's genome. So the conclusion here is the metagenome studies offer an unprecedented opportunity to explore about bee microbiome's community, organizational, and function. It can also form the basis of genomic studies, linking the phylogenetic and functional relationship within the system and environment, which includes all honeybees. It also ensures a more accurate taxonomy and functional categorization of identified sequences by providing a huge information about the existing species efficiently or even newly recognized species in the environmental samples that's related to the honeybees. So here, the experimental design for this metagenomic study of green honey includes, firstly, the honey sample collection is done by collecting honey from those honeybee farms from Malaysia. Next, this honey will be used to carry out DNA extraction and metagenomic analysis. After that, ion torrent sequencing is used to be analyzed all these samples. Lastly, those statistical and bioinformatics analysis is done to study all those related objectives to make sure that the outcome can be achieved. The expected results here are the DNA and the full genome library of green honey can be constructed, and the honey microbiota of different kinds of honey can be identified to study the relationship between the composition of the core microbiome of honey, the nectar, pollen, and the honeybee. Next, to determine the geological origin of green honey. Last, to identify the gene that codes for the green pigments in green honey, whether it is from chlorophyll, algae, or other substances. From this Gantt chart, we can see that about 1.5 years is used to carry out the study of the metagenomics of the honey from June to August of 2021. So the literature review needs to be completed. Next, from September to December, about 3 months is used to collect and extract all these honey samples from Malaysia. And about 2 months is used to carry out the DNA extraction and the sequencing. About 2 months is required to done the proposal writing after all this experiment has been done and the result has been collected. So the proposal writing only can be proceed. And lastly, in May, so the proposal submission can be done. Here has been shown the reference that's been done to carry out this report proposal. That's the end of my presentation for this report proposal of the study of metagenomics of honey. Thank you.